All right, so let me share the screen. Okay, so let's just get started then. All right, good evening, everybody. Welcome to MBSI Bioinformatics Workshop 2. And in today's workshop, we're going to talk about something called sequence alignment. But before that, let's just do some quick revisions of what we learned last week. Hope you still remember what is sequence. Usually, we have a biological sequence, such as DNA, RNA, and protein or amino acid sequence. But usually, we can't really directly obtain useful information directly from them. So we usually want to transfer or store them into a computational method. So we have this sequencing technology, such as Illumina and PacBio, as the way to transfer biological data into the computational data. But after that, we still need something to do something to really help us to digest the information and for further analysis. And that is one of the techniques that we're going to use is the sequence alignment. So what is sequence alignment? Basically is you want to align the sequence that you obtain from the lab and compare it to a database. Usually this could be done easily. The database can be obtained from like NCBI or some other genobanks or other database that you could find online. In other situations, that you might just want to compare two sequences together, like even they're both novel and you just want to compare the similarities. And the Wikipedia definition, or I say scientific definition of sequence alignment, is a way to arrange the sequence to identify the regions of similarity. So it's really for us to understand how similar and how different the two sequences are, or many sequences are. It can be used to study the functional structure and evolutionary relationships between sequences. In the next workshop, you will learn something really interesting, and it's called phylogenetic trees. And of course, some other very interesting applications of sequence alignment, such as like to find out where does COVID from, right? I guess everybody hates COVID now, so if we want to study where does it actually from, is it from like a bat or from chimpanzee? I don't know. There are a lot of studies around it. So first, well, alignments, of course, we have different types of alignments. And firstly, we're going to talk about something called pairwise alignment. Pairwise, of course, is just two sequences together, so in pair. So like, let's focus on some terminology first. I'll give you this question. So we have two sequences now. One is ACTCG and another one is ATTC. The question is how am I supposed to align them? Of course, there are probably unlimited solutions. It's like infinite solutions to this, because if you want to introduce gap, or like you can actually have so many different options to do it. Here, I just want to present you one solution first and introduce you to those terms that we usually talk about in alignment. Okay, so this is the sample solution here. Obviously, we have match and we also have mismatch. And the blue thing here is called gap. But biologically, the reason why gap exists is because we have something called insertion or deletion, if you still remember those basic terminology from genetics. So we actually call insertion and deletion indel, and indel is actually equivalent to gap here. And gap is the result of indel. We have two sequences. Yeah, this is the terminology here. And we have two sequences, right? So we usually call one of them query and another one reference. And the reference, as I just described before, is usually the thing in the database. Or, of course, it's because like you obtained this reference sequence previously, and query is something you probably recently obtained from like lab or from your experiments. That's why it's called query. You might keep hearing this word in your worksheet. So it's just to clarify them. Okay, so after so as I said, there are so many options, so many solutions to that previous question. We do really need something to help us to figure out, okay, well, what is probably the best solution? We call it alignment score. We use this scoring system to really help us answer that question. Which answer is the best and why? Those are just some like random examples that are provided. Here, we provide you like 2 different scoring systems. Remember, we have those 3 scenarios, match, mismatch, and indel. You can literally assign different numerical values to it. Like in the first situation on the left, we say, oh, match is 0, mismatch is 2, and indel is 10. So in that scenario that you really want to minimize the score, because match is the ideal situation that we want, is 0, and all the other situations are like something. Therefore, you want to minimize the score. And oppositely, you can have this maximization system as well. Match is 1 and mismatch and indel, they're like 0, they're nothing. Another terminology here I want to introduce is for the left scenario that like we have mismatch and indel, they are assigned to some like really large or different numerical values. We usually call that the penalty. You might read this or heard this word like somewhere else. If you say, oh, penalty score, that actually means like mismatch or indel. And how heavily you want to penalize those, it really depends on you. For example, if you don't really want indel to happen, you can actually say indel equals to like 1,000. That's possible, but usually nobody do that, to be honest. But yeah, it's just a way to demonstrate those scores are actually designed by you, depend on the situations that you want to use those systems. Yeah. So this is the scoring system. It's a very brief introduction. And now I really want to talk about this sample here. We have this best solution ACTCG aligned with ATTC, right? But how it actually aligns. So this is how we actually do it in practical, in algorithm way to solve this problem. So you have these 2 sequences. This black box down here is the scoring system that we just talked about. You usually write them in this matrix form. And I did this in Excel, so it's just easier for me to demonstrate, but you can just write it on the paper. Like if you look at this table, you notice this gap here. The reason why I write this gap in the front is because, well, you allow this gap to exist before it. Because we never know, like we're not sure if A is actually the beginning of this sequence, the biological beginning of this sequence, right? So there might be something else in front of it. So we don't know. So we just write a gap here, and this is the reason why we have gap, and all of the others are just following well, sequence. The sequence 1 is in a row, and the sequence 2 is in this column. And and the reason why you can see this -A, -16 is literally because we assign -8 to this indel. So if you look at the second cell here, well, obviously it's a gap with A. So gap with A, remember the terminology, is actually called indel. So it's so we actually use this -A here. But why is -A here? It's because the initial value before is gap to gap, which is the initialization of this whole matrix. We say it's 0 here. So if we keep doing this simple math, 0 minus A equals -A, and then -A is -16. And if you keep doing it, this first row and the first column are the initialization of this whole matrix. So what we're going to do next is the real part of the analysis. But before that, I want to talk a terminology called dynamic programming here. It's a little bit confusing if I just explain the dynamic programming to you, but You don't really need to understand how it actually works, but it just gives you a feeling of what it is like. The dynamic programming is just kind of a computational way to solve the problem. This is the thing on the top here is actually the way you write the code, basically. It's pretty straightforward and simple, but the logic behind it is very complicated. A very, very typical sign of dynamic programming is this max here, or sometimes you say min, M-I-N. Basically, it says you choose the maximum value from the following 3 scenarios, scenario 1, 2, 3. And what are the 3 scenarios that we analyzing here in these alignment situations? Remember, like if you remember the terminology, match, mismatch, and indel, like, oh, but it's different here, right? These 3 scenario is if we look at this first cell, you actually have 3 ways to obtain the value for this green cell here. You can obtain the value from the top cell, or from the left cell, or from the diagonal cells. There are actually the 3 ways or the 3 paths that we want to analyze for this, if we really want to fill a value into this green cell. Obviously, A with A is a good match here. The score is 4. So let's calculate the result from the 3 different paths. 0 plus 4 is 4, and minus A plus 4 is minus 4. Obviously, the maximum or the best result is from 0. It's 0 plus 4 equals to 4. So we literally write down 4 in this green cell, and we kind of take a note, say, okay, it's from 0. It's from this diagonal cell here. So if we keep doing that for every cells, well, this picture is just to demonstrate the next cell, and this red highlighted part is just to show you the pathway and where did you obtain the value. If you keep doing this for every cells, right, you will get something like this. Those red arrows are actually the pathway or the path for the of where did you get this value. So the whole path will actually start from the top left way to the down right. So this is like a diagonal path. And this is how this algorithm works, because you literally want to align everything together. So from the beginning to the end. And as you notice, there are a lot of branches here. Well, we literally just to tell you there might be some like equivalent solution. It's not like just one unique answer. There might be some equivalent same answers to it. But the final path, it has to be something connected to the final cell here, the 12. It cannot be this 2 or 6 or A, because that means you ignore some of the C and G or some other nucleotide in your alignment. So this is just a very, very brief introduction to how the scoring system could be used when you do the alignment. And the algorithms itself is not really it's not really the thing that I want you to remember. It's just to give you the feeling of how the scoring system could be used to evaluate how we're actually going to do the alignment. So that's all about nucleotide. So what if we want to do with something like protein-based, like amino acids? We need something called scoring matrix. And this matrix, okay, let's jump into this picture first. So obviously this is a matrix. And this matrix is literally pre-built for you to use directly. You can just import it from the database to use it. And the thing I really want to highlight here is you may notice like even A is matched with A. The value is 4 and R with R is 5. They're different. So all of those this diagonal line here, the value are different. It's because, well, for nucleotide, right? If ATGC matches with ATGC, that's very straightforward. But amino acids, they have this codon system and they have this like historical, evolutionary, and chemical, biological properties. They're all different. So the weight of matching are different. So if you see this sequence, say A with A, it's probably different or weight differently than R with R. So how do we demonstrate those differences? We just assign them to different values. And the takeaway from this is the higher the value is, the better the outcome is. So if you see something like C with C is 9, which means this is a really preferred alignment in this case. And obviously there are some like negative values and 0, but that's just related to the mathematical way how they calculate those matrix. But in practice, you just need to import this matrix and use the value inside of it and for your evaluation of the outcome of alignment. And there are two commonly used matrix called PAM and Blossom, following with some numbers, but those are all related to how did they actually calculate this matrix. But you just need to remember like there are two like very commonly used one, PAM and Blossom. Okay, algorithms again. Well, the way I described how we do this alignment is actually the algorithm itself. But here, what I'm going to say is the classification of algorithms. There are many types of pairwise alignments. What I want to talk about is these two, global alignment and local alignment. I really suggest you to have a look at the picture, the colorful picture here. I think it's a really good demonstration of the difference in between them. So the global alignment basically is we assume that the two sequences are at like similar lengths, so that it's basically you want to compare them together like globally. But local alignment is you assume one sequence is only a part of the others. So it clearly demonstrates here that it's just a part of it, and you ignore the end. So you don't really initiate those gap for it because you know this is just a part of the reference. So you don't really care about the others. You only care about a part of the local, part of the reference. There are a lot of algorithms like Needleman-Wunsch. This one, Needleman-Wunsch, is actually the algorithm that I was talking about here in this matrix. This is actually the run through of the Needleman-Wunsch. There are also others like Levenstein and Smith-Waldman. It's basically saying, you could maximize or you could minimize the score. And we just call them like, different names sometimes. Well, this is like how you name algorithms, right? But the name is just a name. Usually when we really want to do this alignment analysis, you go to the website, use the tool, and you just directly choose one of them. It doesn't really matter like at a beginner level which one to use because they usually provide you similar outcomes. But if you get to the advanced level that you might need to understand for a specific case, what kind of algorithms you really want to, or it's slightly better than the others. But for our workshop, I think that's too much to talk about. Yeah, one thing here is very important is called BLAST, is a part of the local alignment. As the name suggests, it's a basic local alignment search tool. And you can find this tool here in this website, MBSI. About BLAST, because it's really commonly used. So that's why we want to introduce you to. And in the worksheet, you have a chance to use it yourself and to feel it, like how it actually works. So there are different types of BLAST, BLAST N, BLAST P, X, and some others. They're just about, they're just to tell you what are the inputs of the BLAST. So if you only focus on nucleotide, then it's N. Then if you only focus on protein, then it's P. If you have one sequence is like nucleotide and another one is the translation of something like the proteins, then it's like X. And yeah. And this is just a picture to show you what it looks like in a very high level way. One thing to emphasize is this e-value. When you look at the outcome of BLAST, you will see the e-value column there. It's basically to tell you how good the alignment result is. So the smaller the value is, the better the outcome is. Yeah, this is the key takeaway here for BLAST. Okay, so that's enough about pairwise alignment. So for pairwise alignment is one sequence to another sequence, right? But what if you have 100, 1,000 or millions of sequences together at the same time? You want to see how good they aligned together, like, all at once. So this is something called multiple sequence alignment, MSA. I really want to emphasize this, right? Pairwise alignment, or all of those algorithms that we were talking about, it's pretty simple because there are only two sequences. You can even do it by hand, like what I just demonstrated, right? You can just draw the matrix. But when it comes to 100, 1,000, million, you can't really handle that. You cannot handle that. The computer sometimes even cannot handle that. Because, well, let's go back. Think about like drawing those scores here. And this is only for 2. And if we add another one and another one and another one, how complicated it is to do all of those analysis. And this is only for like 4 nucleotides and 5 nucleotides. What if the sequence is like a million, like 1,000 BP? You can't really handle that. So we really cannot use dynamic programming, this method, anymore. Well, the dynamic programming is really to find out what is the optimal solution. Like, of course, if you're dealing with multiple, I mean, a lot, you can't really find the optimal solution locally. So maybe you need to like sacrifice for some of the optimizations locally, but to achieve these global optimizations in like for all of those sequences together. So we have this method called heuristic method, is we introduce probability into consideration. So it will kind of provide us like generally acceptable answer, because we don't know if it's really the optimal solution, but we consider it's like highly likely to be the best answer. So we just accept it, right? Because based on the current computational ability the human being has, right? We can't really solve that kind of those complicated problems. So we say it's a heuristic method. Why do we need to do those analysis anyway, right? Because we can literally use those studies to focus on like to do some analysis of the homologies, and of course find the COVID, find virus, a range of virus, and also we could do these things called phylogenetic trees, which you will learn in the next workshop. I think that's a very interesting topic. So I do suggest you to come next week and learn with Jack and see how it actually work. At least it's some real applications that you can use. This is just a picture to demonstrate the outcome of it. So you can see those, like they're all color coded, and there are a lot of similar region, and there are a lot of like variants. So these small variants may tell you something biologically, and those conservative area may also tell you something. And well, this is actually a good demonstration to see. So we really want to achieve the global optimization, or the global best solution of it, rather than a local best solution. If you look at those the RLA0 and this PLAF8 and SULAC, those two, obviously like they're not locally aligned really well. Probably there are some better solution for it, but because we're putting it in this big data group, you have to sacrifice those local optimizations to really satisfy the global one, right? This is just to emphasize like why we cannot use dynamic programming here. Yeah, basically you will get something like this. And it's pretty interesting. And of course, there are a lot of future study or further analysis that you can do, like a variant calling to tell, is this really a variant or it's just a reading mistake? Okay, there are a lot of studies related to that. But yeah, for our workshop, that's a little bit too much to talk about. Finally, I'll just briefly touch on the algorithms for MSA. Basically, three things I want to say: the progressive method, the iterative method, and the probabilistic method. Well, the progressive method is thinking about like you're building a tree, right? Like you choose, so you do pairwise first, but you do pairwise only to those pair that you think is the most close to each other, the closest to each other. Then you keep adding the other pairs and to it. You like building a tree from the root to the top to the branch, and that's why it's called progressive. Those tools like Cluster W, T Coffee, those are progressive methods. And iterative method like muscle. It's not really like the muscle. I mean, it's a MUSCLE, is muscle, we call it muscle. It's an iterative method. It literally is a kind of like improvement or like better solutions maybe compared to the progressive method. Is like so you don't just like after you do the first pairwise alignments, you don't just like leave it alone and never use it again, but you keep realign those initial sequence to others. That's why it's called iterative, because you introduce this iteration method in it. And the probabilistic method, or well, the most commonly used one is called the hidden Markov models. And this model is like, you know, it's introduced this probability method into it. Those are just a name to you for now. Yeah, and again, like when you really want to do some practical play about MSI, you literally just need to choose one of those algorithms on the website tool or on other different tools that you just choose the name of it. So that's why I want to introduce those names to you. So when you see them, you know, okay, this is the MSI, or this is a progressive method. Okay, and that's literally everything I want to cover in the lecture part. We have this worksheet available now, I think. Yeah, so I think it's your time. If you have any questions, feel free to ask. Stop sharing. Okay. I don't think we need to go to break.
Yeah, I was thinking that. I was starting to sign up and I was like, oh, actually, yeah. Did you stop recording?
Oh, yeah. Stop recording.